I'm happy to introduce our speaker, Juli Petereit. Juli has a PhD in biomedical engineering from UNR, and she has a master's degree in applied math and a bachelor's degree in math, and she now works at UNR in the bioinformatics biostatistics core as a bioinformatics analyst. And so I'm going to turn it over to Juli.
Thanks, Laura. And then we're switching to the presentation.
When you want to use the slides, if you want to introduce yourself or something first, just holler at us and we'll switch back.
I think I will just get started with the presentation, and then we can switch back to me. I'm not that important.
You're not that important.
No. Can you hear me okay? Everyone?
Yes, we can hear you.
Yes. Okay, thank you.
It took a while to switch last time, right?
I couldn't get it to switch at all.
There we go.
Oh.
It's just slow.
So I will talk about a little bit today bioinformatics, what the Nevada core does, and how I got into bioinformatics, and then we'll give you a little bit of taste of biostatistics. So just to start, I will introduce our team. Karen Schlauch is our director. She manages everything and pulls in the projects and tells us which projects we're going to be working on. Richard Tillett, he presented at TMCC in February. He's our sequencer. He knows everything from computing to the real in-depth science. He's our scientist in the group. I am a data scientist, more coming from the mathematical side. As Laura already said, I have my master's in applied math. Alberto Nava is our undergraduate. He's working on an RNA-seq pipeline under the direction of Richard Tillett. And Joe is our newest member of the group. He is going to coordinate workshops in all the community colleges around Nevada and is going to do the outreach at Nevada INBRE Bioinformatics Core. So this is our team, just to give you an idea how many people we are, and we're not all full-time. So an overview of what I'm going to be talking about in general is what is bioinformatics, what does our core do, a little bit more in specifics, then how to engage into bioinformatics if you're interested into it, what are the steps that you can take to move towards that direction, and then a little bit of biostatistics. And please interrupt at any time. This is a presentation, but I want it to be interactive because sometimes when you wait until the end for questions, you completely forget. So just raise your hand or interrupt and let us know if you get stuck or want some clarification on some of the things. So bioinformatics is the science of collecting, organizing, and analyzing biological data using computers. It's a pretty general thing out there. If you go out into the desert, collect some rocks, you look at the rocks, you specify them, you write something down, you put it in an Excel spreadsheet, and you start analyzing it, that's bioinformatics. You are doing something analytical, you're analyzing your data, you're collecting data, it has something to do with the sciences. So it's really broad of what bioinformatics is, and it's very overlapping with other disciplinaries. And one of the main ones are life sciences, computer science, statistics, and math. And any bioinformatician can come from any field. Some know more about another. So as I mentioned, Richard Tillett is certainly our life scientist. And he took on all of the computer science by himself and learned it later on, and he is super efficient. But if you would ask him, like, oh, are you a computer scientist? He would never say yes, because he doesn't have the background in that. Where Karen and I come from the mathematics side. Karen has her PhD in math, so she is really analytical and very strong in biostatistics and statistics and the pure math behind it. So we both come more from the analytical, mathematical, computational side, and we picked up the computer science as we went on along too. Again, I wouldn't consider myself at all a programmer, but I do program. So if I share my code, and that's always something really scary when you start sharing your code with others, because everything should be open source, so everyone has the resources to use, and you don't have to start from scratch every single time. Like, that's academia, right? We should be collaborating to get the best out of our research. So it's embarrassing. Because if a computer scientist looks at my code, I'm embarrassed, but that shouldn't hold us back from sharing. We all make mistakes, we all start somewhere, and sometimes you find a great collaborator and says, hey, you could be improving your code doing this way, and you have a new colleague. I mean, that is pretty exciting too. So don't ever be embarrassed of your work or code, because it is actually very scary when you start out. So also bioinformatics doesn't stop. It's not math for the most part. When I took math, you can pick books from the 1850s and it's still true today, right? You can pick a book from 1970 and it's still a very good book. There are some new methods that are still being developed, but all the old principles are still true. Not necessarily in science. Something that was found 100 years ago has been redefined and clarified. So bioinformatics is the same thing. It is very fast moving. It does not stop. You have to be on top of things all the time. You have to continuously read papers on what new methods. In the last 2 decades, or 2 decades ago, microarrays were the big thing to analyze your genomes, and they have kind of died out and have been taken over by RNA-seq, a newer method to analyze your genomes. Well, if new methods are developed, the technology changes, your data analysis needs to change too. Also, the computing power is constantly changing. So if you are in bioinformatics, you have to be open-minded to new things. You cannot be set, as you will all get, like if we do operating system, right? We have new Windows. Windows 97 was great, and XP was like, oh no, this is so different. And then 2010 is like, well, where are my buttons now? So there is some frustration that comes with it, but also the new operating system gives new tools. And it's the same thing within the science. Like we get new tools, it's a bit more exciting, we can go deeper, we can go in more depth, but we have to be willing to be frustrated for a while, and then get excited again with the new tools we have. So that's kind of the scope of what bioinformatics is. It's constantly changing, it uses life science data, and the life science data, as I mentioned, can come from anywhere. And then we analyze these data and try to make sense of it and send it back to the scientists, because we don't necessarily understand the results from the data we get, for example. So what we do at the Nevada INBRE Bioinformatics Core is we work mainly on omics-focused projects. That means that we look at an entire system at an instant time period, or we compare 2 time periods of the entire system at once. We don't just look at one little pathway within a system. We want to know it all at once. So we focus on genomics, transcriptomics, proteomics, and metabolomics. So all these different data types. So the genomic is genes, clearly. The proteomics are the proteins. So they do relate, but there's also some difference. So if we want to know everything about a system, we cannot just look at one omics area and forget about the other. So the goal is to integrate them all at once to get the most of our research. It is not easy to compare them because there's not a one-by-one mapping where we can say, oh, this genomic data corresponds to this proteomic data one by one. It has to be always a lot of extra work that needs to be done. But bioinformatics is more than just omics. If you're very interested in pathways, you can be more specific. It's just our core is more omics oriented. We are more of a systems biologist, if we want to talk about it that way. And then we'll talk about a couple of classes that UNR has to offer, and then we do offer workshops once in a while. For example, the ones we have been hosting at TMCC. We also had one at the University of Nevada, Reno 3 weeks ago about R programming. So we try to put out workshops here and there as we can, and time allows us to. So one of the things is part of our big work area is working with DNA. So what's being done is DNA is extracted from some organism. What the organism is dependent on the researcher we're working with. And then this organism needs to be broken up in smaller pieces. The DNA itself, like for example, the human genome has 3,200 million base pairs. So to look at the entire genome, a sequencer cannot take the entire thing. So you break up your genome in 1,000 little pieces so you pretty much, let me rephrase. You take your genome, you extract your DNA, and then you have your DNA, and then what to do with it. And then you start smashing it up. So I really like this picture. It's one of my favorite quotes from a cartoon. Does anyone know The Emperor's New Groove? It's like, I'm gonna put him in a box, and I'm gonna put this box in another box, and I'm gonna smash this box. No, I'm gonna smash the box with a hammer. So I just think that was a pretty good, so you can see that this will be your DNA, and you're gonna smash it down with a hammer, and then you get thousands of little pieces. And then these little pieces can then be fed into the sequencer, right here. And then that sequencer will evaluate, have some biochemical components, it looks at the DNA, stuff I do not understand because I'm not a biochemist. So the sequencer does the trick, looks at all the base pairs, turns them around, and gives them digital values at the end after it sequences through. But now it only sequences these tiny little pieces you started out with, that you fed into the sequencer. You don't have the genome at that point. So then you have these thousands of little puzzle pieces, and you're trying to put it back together to go back to where you started from, that resembles your organism again. And some organisms are nice. For example, the human organism, we have a reference. So we know what the genome looks like. So you can think of that you actually see the colored pieces of your puzzle, because you kind of know which piece should go where, because you know, you have that picture in front of you where it should go. If you pick an organism that you haven't studied yet, something completely new, a plant, a little bug, something that has not been sequenced, then you could think about it that you're looking at the back side of the puzzle, that you have no idea, every piece looks the same, other than that the edges are different. So you're really just matching the edges, so it takes much longer, right? It takes way more computational power to be able to put that genome back together so it resembles something we want to look at. So that's the difference between aligning a genome and assembly a genome. Aligning is when you have a reference, you see the puzzle pieces, the color pieces, and assembly is when you just have the back part of the puzzle, which is a much harder problem, and JR works on both of those, Richard Tillett. So that is really exciting. Again, I am not working on aligning and sequencing because that is a field where you have to understand the science a little bit more again. So for a pure mathematician or just a pure computator, that problem will be extremely hard because we don't understand as much about the genome as, for example, Richard Tillett does, because he has all that background in biochemistry. So someone like me could take it on, but I would have to learn a lot to come to the place that to be efficient and sufficient at it. So then after we have them back together, we have our puzzle piece done, we then need to go back in and see what can we find out here. So we converting the genome, what we see right here, with the ACTG, and see what else can we identify. What else can be pulled from there? So from these aligned genomes, we can see what genes have been expressed more based on how many alignments we got, for example. So that's how we get gene expression data. And then we can go in and look for a testing hypothesis, something we're interested in. And normally these testing hypotheses depend, or the experimental setup should depend on the experimental hypothesis we started out with. So if I'm interested in a drug and its effect on what genes are affected by it, then I will sequence or take a sample before I apply the drug to the tissue or an organ or just an entire human sample, and then I apply the drug, and then I take the sample again. So then I look at the difference between the two genomes, or the gene expression in other words, to see which genes have been affected by the drug, for example. So this is the assembly and aligning. What I mentioned before is pretty much all Richard Tillett in our group, and he worked, for example, on 2 different pine beetles. He did both of the first aligning, the mountain pine beetle and the Jeffrey pine beetle. I was really excited. The mountain pine beetle, actually the paper just got accepted and will be published soon. So that was exciting. And then 2 Clusia species that he assembled for the first time. And that's still being written. So it's really exciting. And what I mentioned with the assembly, it costs much more money, human labor and compute time, because you don't know as much. So it is much more costly compared to if you work with a genome that you know something about. Working with a model organism, just lost the word. Like if you work with a model organism, there are tons of resources out there. There are tons of databases, there is references, so you're not as lost. You kind of have a guidance on what to do with a model. Then you have also, let's say, pineapple in the plant world has been getting more attention, so there's more and more out there for pineapple, but it's still not a model organism, but at least there is something. And then you might be stuck with some beetles that you want to learn about because you don't want them to take over our forests, but you have to start from scratch. So there's a lot of work. Is there a question?
Yeah, from me. From GBC Program here, this is Rita.
Hello.
I would like to ask you, why did you select the beetles or this 2 Clusia species? Was there anything special about this beetle, or are they parasitic or something?
Yes.
Thank you.
Both of these beetles mass attack trees, and they're devastating to forests, mainly in Colorado, Canada, and somewhere else in the Midwest. And Klaus Tittiger and Gary Blomquist at UNR work with these beetles, and they have been trying to figure out the pheromone pathway within them to identify which pheromones the female give off, so the male will. So the females call the males the moment they give off the pheromones after they start feeding off the tree, and they want to figure out that pheromone to manipulate it so the male will not mass attack the tree together. The Clusia is part of a DOE project for biofuels. So I'm not 100% sure if Clusia is defined as C3, a CAN plant, or C3. I'm not part of this project as much, but they're working to figure out the CAN pathway within these. So CAN plants are drought, stress tolerant, salt, sun. They also don't need to be watered as much, so they're trying to figure out how to model that pathway within these plants into other C2 plant families to be able to have crops in Nevada, for example. So we can plant them out in the desert without having to worry about watering them. So those are the main reasons for those 2 projects. Clusia is part of John Cushman's and Karen Schlau's DOE grant. So why doing RNA sequencing? So the assumption is that the gene expression that you get from these sequences give you an idea about the biological intent. And if you talk to really biochemist that works on protein, there's like, you're wrong. How can you ever assume that gene expression will tell you anything about the protein expression? So there had been a lot of controversy about our gene expression correlated to protein expression, and it's not always true, but at least it gives us a starting point. If a gene is very active, it has a meaning, right? So we can figure out then later on, and can follow up the RNA sequencing with proteomic studies if it's really true that these proteins are affected, what transcribed for this particular protein. So it's a starting point. And then also what's really nice is we get thousands of measures with RNA-seq. We have a big amount of data we can analyze in many different ways. For example, Corinne Kramer at UNR again, he's working on wine grapes, and he has a huge data set of a couple of white wines and red wines. He measured them at different sugar levels of ripening, and that data set is so big that he has published 2 papers already, and he's a 3rd one, and he's still using the same data set because you have so much that you can look at different things. And as I said, you find one thing and then you can follow up with a metabolomic analysis or proteomic analysis to cover your hypotheses that you got from the RNA-seq. Let's see, another thing that the court does is genome-wide association studies, and these are used to identify markers and correlate them to specific traits or diseases. So you look at an entire population and you look at little SNPs, singular nucleotide modifications, and see which ones are uncommon. And then these SNPs can be hypothesized to be markers. And if you do a genome-wide association studies where you look at a greater population, you can statistically back it up by your statistics that this actually might be a biomarker. And if you find a biomarker, that can then be used for drug development or gene therapy, whatever the scientist decides on how to use that biomarker or how relevant it is. And that is something that Karen Schlauch works on intensively. Biostatistics, we help with setting up experimental setup. So also before you actually start, it's nice to talk to a biostatistician, to talk to a bioinformatician, to make sure that your sample size is sufficient, or that you're actually sampling what you're hypothesizing for, that the math or the stats behind it will be able to answer the question you would like to answer. Most of the time, the biggest problem is sample size. If you collect 2 samples, that's not really going to get you anywhere. You need replicates to make sure that the measurements you get are not just artifacts. Then it also includes some data quality control. As I said, after you sample, you experiment, you collect your data, there might be some collected data that are not good, that something happened, that plant maybe got too much water, or if you talk about a human sample, maybe that person had a cold during the test period, so it messes with your data. So it needs to be data quality controlled before you move on to analyzing it. Because if you throw anything at the analysis and don't look at what you throw at it, what you're getting out will not be relevant, right? Because it might just be artifacts within your data. So it is important to go through and look at your data before analyzing anything. One of my favorite examples is I got a metabolomic data set, and they asked me to analyze it. And because they've already worked on it, I didn't spend too much time looking at the data. So I started analyzing it, and right away I noticed something was off. It's like, there's something not right here. And then I started looking at the data, and then I had about 20 metabolites that had the same measurement all the way across all conditions. And it's like, well, this is wrong. That is just not possible. And then I went back in and said, can I please have the raw data before it has like transformed? And they imputed the data. So somebody thought, it's like, well, this one metabolite had one measurement over 20 measures, and I'm just going to impute the rest, pretty much making data up so it fills out the empty spaces and move on. This is completely made up data. You really in my point of view, you should throw this metabolite out. If you have one measure out of 20, you're not going to get anything out of it. If you, let's say, if you have 10 measures out of 20, it's still not great, right? You only have 50%, but let's say you have 10 measures under high water conditions and 10 measures under drought conditions, and these 10 measures are just in one of those categories, that's different. So if you just do a percentage of what data is missing in each row, that doesn't really work either, because those 50 that got measured in the drought stress, for example, are really important because they weren't measured in the high water condition. So it is important to get a feel for your data before analyzing it. And I cannot talk too much about it because it's a really common mistake. So you don't want to blindly impute your data. You rather want to throw some data out. Sometimes it is necessary to throw out data points even if you don't want to, just because they are garbage and you shouldn't use them for your analysis. And then we support projects including, one particular example that we're working on is the effects of poverty on infant mortality rates. So this is a really clear biostatistics. It is not so much bioinformatics, but this is very a biostatistics project. And then another thing our group focuses on is high performance computing. So we want to do our analyses, our alignment, our sequence analysis, assembly as fast as possible. And so to do this, we have 2 computer clusters and that have a total of 500 CPUs, about 7 terabytes of RAM, 96 terabytes of storage, and 10 gigabyte network. So we can parallelize the alignment over multiple cores so we can speed it up. And that is one of the projects I mentioned at the beginning is Alberto, who's working on the RNA-seq pipeline under the direction of Richard Tillett. It's actually really exciting. And then we go out sometimes and do these workshops, which is really fun to meet other people and collaborate and hopefully come up with future projects. We support grants. Right here. We also help writing them. If you're at a loss where you have a really great idea but you don't know what the analysis would be, Karen and Richard are the ones that work on that the most, to help writing grants too, to explain the experimental design and what the analysis will be down the line. We give workshops, and then what we started last year, we started giving out little service awards to support smaller projects, which has been really exciting to be working with students and some starting faculty. So here's kind of a graphic of what our daily life looks like. So another thing I wanted to point out as a bioinformatician is we're not the scientists. We cannot really interpret the data as much. But we have to be able to present the data to the scientists in a way that makes sense to them, not necessarily to us. So I don't ever work in Excel because I think Excel can mess things up, right? Like changing a number to a date. It's like the common thing that I just get so flustered by. It's like, don't change my 0310 to March 10th. Like, just leave it at the number. So Excel can be somewhat error prone. So I wouldn't be working in Excel, but the scientist does. So when I give my data back, I have to give it back in a way that the scientist will be able to understand it. And that includes data transformation, and then also making like fun little graphics. So I just thought it was kind of fun to see where we spend our time. Clearly, next generation sequencing is the one we spend most of our time on. And we still do microarray analysis. Although they're getting old, they're still great. Because if you have a model organism or a well-studied organism, you know what you want to look for, you can make an array with just this many genes that you're interested in, and you can sequence it. It's much cheaper, it's precise, it's not necessarily a whole entire omics approach, but you can still classify the systems if you have a lot of genes on your array. GWAS is the genome-wide association studies that I mentioned that Karen works on a lot. We do a little bit of it. Network analysis, that's mainly me. Then the pipeline development is all of us, really. We're all put our collective knowledge, because we all have something else that we can bring to the table, right? So JR, Richard Tillett, brings in all the alignment sequencing. Karen Schlau does a lot of differential gene analysis, which I'm going to mention in a second, and I do a lot of in-depth network analysis. So we all bring something else. We're all part of this pipeline development. So it's kind of fun. It's never always the same, and it changes up, and then something else comes in. Last 2 weeks, I looked at some proteomics data, very different, very scientific, and it scared me a lot, but you have to be open to new things, so it's fun. So engaging in bioinformatics, how can you get there if you're interested into it? So these are just classes at UNR, which I know doesn't apply to everyone, and I probably should have put the titles of the classes more than the names. But for example, UNR has 2 bioinformatics classes, the biology 47607 and the biochemistry 709. There are the only 2 classes that have the word bioinformatics in them. And both of the classes look at a lot of the databases that are available and publicly available methods for both classes. Clearly, the biochemistry bioinformatics class is a little bit more towards graduate students. You can already have a project and tell that's what you want to work on, and then Karen normally adjusts the syllabus to make it applicable to the students that are taking the class so they get the most out of it. And the 407 is more of an introduction, like these are the tools, that this is what you can do, this is the direction you can take if you're interested. But as you can see, all these classes are in different departments. The math class, the 420, is a math modeling class. It just kind of gives you an idea on how to model things. If you've never used math to solve real-world programs in a little bit bigger sense than calculating, oh, what should I pay in tip? This is a great class because I don't think it is too much math. It really gives you more of an idea of how can I create a model, and if I have this many pigs, this many sheep, and I want to figure out how often I have to buy alfalfa and different things to feed. And so you build these kind of fun models. I really enjoyed this class. And you do a little bit of programming, and it's not hard. I mean, it is hard because you're just starting out doing it, but it is not the in-depth where you have to do a lot of back-end like deep coding. There are functions, and you have to call the functions in a certain order, and you maybe have a little for loop once in a while, but it's not too hard. The AP ST classes, I think they're great too. They're applied statistics classes. So they're not as math-based compared to the stats classes that are often in the stats department for statistics. Then the CHS classes are the same. There are community health science statistics classes, so they also are very applied, although I think these are more, the APST stats classes are more general. Then there are computer science classes. The first 3 are really just the standard ones. They will teach you nothing in particular. They just teach you about coding, how to think about to write a for loop, or what should you do if you want to multiply row I and row J, in what way. So they just really talk about the algorithm and the thought behind programming. Then there's biomedical engineering. They just have a couple classes, and there's introduction, and they do talk a little bit about bioinformatics, and it just gives you an introduction to the field. So as you can see, all these different classes are pulled from different departments, and that's what bioinformatics is. You bring one strong background And that is your background, and then you just have to add a little bit more knowledge from other fields to it, but you don't have to be an expert in these other fields. So I talked about only 2 classes, but that shouldn't stop anyone to look in that direction. Another way to figure out what you like about bioinformatics, or if that's what interests you, is reading papers. There are tons of papers out there all the time, and if you just put in bioinformatics and then your organism of choice that you're interested in and what they have been doing, you will find a couple papers. I've read tons of papers to figure out what I really want to do. And what is part of bioinformatics, no matter what, there will be some programming. There will always be some programming. If you completely want to stay away from programming, I think you could move more towards biostatistics, but even biostatistics now, because the data get bigger and bigger, you have to do normally some programming to parse through your data. So some of the programming language that are really hot in the field are Python, R. There is still some Perl. Ruby is getting stronger again too, and there are many others, but those are just the ones that I personally think are the strongest. Richard does Python and R and Perl. I mainly do R. I do a little bit of Python. So also sometimes one programming language is not good for every task. So you might pick a problem that is much better in Ruby than it would be in R, just because you maybe have some graphical interface in there too. So it really depends. Also, Perl is great for data parsing. If your data is huge and then you have to cut it down, you have to pull certain rows first before you want to do any analysis within R, for example, you would do it in Perl, because it would take so much longer in R, and what can be done in Perl in one line would take 5 lines in R. So it also depends on what your problem is to use what programming language. Sometimes it is actually rather hard to figure out, or you figure it out by talking to someone. You're like, oh, I've been spent 2 weeks solving this problem on R, and they're like, oh yeah, you could have done this in Python in a day. So it's like, oh great, now I learned something in those 2 weeks, but next time we'll use Python for that problem. So again, talking to others and not being afraid of showing code, I think, is one of the biggest lessons I've learned over the last couple of years. Also getting a little bit familiar with command line. It is extremely uncomfortable at the beginning. I will talk about myself in a second. I was not a big fan when I looked at it the first time, and I stepped away for 5 years and didn't touch it again. And then I went back and was like, ooh, this is actually kind of fun. So sometimes it also takes a little bit of growing up on my part, for example, or just to be open or take a workshop, because if you're just looking at a black screen for command line, you get overwhelmed. It's like, I don't even know what to do, or where to start, or how to even deal. And Ubuntu is a very nice back end now. It's not as scary anymore as what I had when I started 12 years ago. So Ubuntu is an interface that makes it look like more what you're used to from Windows or Apple. So that makes it just a little easier and less scary. And then stealing code from other people. That is sometimes very beneficial, or it can bite you in the bottom too. So you want to understand at least a little bit of what the code is doing that you're stealing. You're not stealing, it's open source, but what you're borrowing. Because if you don't know at all what they're doing, it might do something completely wrong or different, or they had a different assumption than what you were having in your analysis and you didn't consider that. So just to have a general idea on what the code is doing that you're taking is very important. And then at the end, as I said, the learning never stops. But when you're passionate about what you do, none of the work seems that bad, right? If you're just doing it to pay your bill and you go home again, or you're doing it because you got this stupid project for a class, you don't learn anything, right? I've taken so many classes where I had to do something, but I was not interested in. Do I remember any of these projects? No. do I remember projects I was so, yes, let's do this, let's get together, let's do some more, let's look at the back end. You get more out of it and you remember that. And I think if you want to look at any of this, you have to be passionate about it because it's not easy. And you will get, I mean, research is frustrating, right? You will hit a wall for half a year and then all of a sudden everything works within a month and you're like, oh, why didn't this work in the first place? So you have to love what you're doing to be, I think, successful and not be just frustrated about your day-to-day work. So that's kind of where to go if that interests you. And then how I got here. Laura already mentioned I got my bachelor's in mathematics, and that's where I kind of started with programming. I thought if I do math, I should do my minor in computer science without really knowing any programming. And I really like the back end of programming. I like the algorithm. I like to figure out how to solve the problem in an analytical way, but I didn't necessarily like the programming part. I like thinking about it, but I didn't like sitting in front of a computer and coding it. But at least it gave me the idea and the logic of algorithms. And then I also did math class where I did the modeling. That was really fun because you start using equations in math and put it all together to solve something in the real world. As I mentioned, my second programming class in the computer science department was command line, and I wanted to drop out. I was overwhelmed. I had no idea what I was looking at, and I think it was a little bit because it was cold turkey. They went from a Windows coding to a Linux coding, and there was no introduction to what a Linux system is. And I think their thing that, oh, if you sign up to take a computer science class, you must be computer science geeky. But there are also so many people now, you need to know computer science where you aren't necessarily yet that type of geek anymore. So I think I need to have a little bit more introduction of just a different operating system. Then I moved on to my master's in applied math, and I did more analytical math and numerical methods for a hydrology problem. So it was kind of, I looked at how the water dispersed in different media and how it interacted. And that was really interesting, but I still wasn't sure what I was doing. And I always thought I always wanted to do math. It was what came easy to me. I'm not a big reader, I'm not a big writer, so the numbers make just more sense and are more fun to me. But I always thought I had to do physics. Math always was, you have to do physics, and I never liked physics. So it was a very frustrating process that I wanted to do math, but I don't want to do physics. So attending conferences, doing actually my bachelor in math helped me figure out what I liked. Just using the math to life science problems. So that took me away from the whole physics dilemma. And I liked seeing, I went to one talk during my master's, they analyzed the path of ants and how the dynamic worked. And I thought that was so interesting. And I just started going to different talks. Did I understand something in these talks? Most of the time not. Like talks at conferences, especially doing your bachelor, still at your master's level, you're just like, wow, this is so above my head. But you get an idea what other people do, and you get an idea of maybe what interests you. So I really think that attending these conferences was one of my best ways to figure out what I like doing, really. And then I came back, and because I knew that, I started out my PhD in biochemistry. And I was in Dr. Christine Cremo's lab for 1 year, and it was just so much science. And it's like, this is just memorization, that just doesn't make sense to me. And coming from math, from my viewpoint, math was just logic to me. As like, there was a problem, figure it out, learn what the math is behind it, but there was no memorization for me personally. I know other people feel different about it. But for chemistry, it's like, why do I need to know these components or where the peptide attach? Or it was just too much. And I talked to Christine, and I took Karen's class my first year, the bioinformatics class, and I was like, ooh, this is cool. I like this. There is some databases I'm not really that interested in, but I like the math and the algorithms behind it. And that's how I started with her. And I took a couple more health, biostatics, community health science classes in statistics. I was like, this is kind of fun. Like, I started stepping more and more away from the math part and did more statistics. But if you have a background in math, I think the statistics comes pretty easily. If you have that logical thinking of solving problems, the statistics kind of goes along the same way. And I started playing in R. So 5 years ago, I didn't do anything in R, and I ended up writing a library. So that was a really fun process. And I started doing command line again. And as I said, maybe at this time I wasn't as scared anymore, or I just grew up, as I said, and it worked. And now that is my favorite operating system to be on. I use Windows to make presentations and everything else on a Unix machine. And then I read a lot of papers and books from the library, which doesn't happen so often anymore because we do have Google, right? We can read everything, but I always think too, if you want to reference something, it is nice to have the original. So I also pulled a lot of books and read through a lot of methodologies that were created 50 years ago and they were reinvented in the 90s or 2000s to apply to real world problems. So that was really fun. And then for my PhD dissertation, I looked at after the alignment, and you get the expression of each gene. These are just numbers. And those are the files I look at. Here we see in the first, we don't see, but in the first row, first column, we have gene identifiers. In the first row, we have the conditions, and then we just have a blob of numbers. And I'm the one that tries to make sense of these numbers. So after Richard Tillett aligns and assembles the genome and takes all the counts into, we get at the end some gene expression values. And so one of the approaches I mentioned that Karen works on a lot is differential gene expression. So she would take this data set and try to figure out which of these conditions are very different from each other. That's kind of the scope of differential gene analysis. You do a 2x2 comparison and see, oh, is the baseline different from the treatment group? And then you maybe get 500 genes for that list, and then you have a little bit narrowed down hypothesis, and you can start looking at 500 genes instead of 13,000 genes. I don't care about differential gene expression when I work. I take the entire data set. And then what I worked on is to take the entire data set and build these nice looking networks. They don't look this nice because we're talking about 17,000 of these little points. So it's just a blob of points really. So we need, or I used then numbers and mathematical models to create this and still have the math behind it. For publication, you always want some nice pictures, but then you would pull something from these 17,000 genes and publish something like this and not a whole blob of numbers. And then what's interesting, you use the entire genome that you sequence, build a model, and you have some math behind this model, and then you can figure out, example, how to get from this gene to this gene in the shortest path. So mathematically then, it figures out which path to take to get to this gene in the fastest way. That's one problem you can solve with these networks. I did not work on that. I was more interested into going inside the network and see if I can find groups that are very highly interconnected. So that means if these genes are connected, that they behave, that their expression values across these conditions are very similar. So let's say all these genes go up, have a really high expression in the first part of the experiment, and then they kind of flatline. So if these genes behave similar, we could hypothesize that they are part of a same pathway, that they share functionality. Again, these are hypotheses. They're just mathematical models, and sometimes people mistreat this. Models are not a proof of concept. They give you an idea of where to go back into. The scientist needs to take this information and thus follow up experiments to validate the mathematical model. Mathematical models don't really validate, they give you ideas and narrow down your hypotheses. So then we see that in this case, we have a very highly connected group. All these genes connect, so that means they all behave similar over the conditions you take. So that's kind of the hypothesis and the underlying assumption of this method, that you find genes that cluster together to hypothesize about their common biological function, hopefully, or being part in a pathway. So that was the main gist of my PhD work. So a little bit of biostatistics. I found this picture and it just made me laugh. I like to create images to just kind of introduce a subject. So I always put into Google image search and I put biostatistics in there, and that's what I got. And it's like, well, I guess that explains it all and I don't need anything else. So really frustrated. You have to make jokes to get through it. It can be really boring at times too. Makes you scream. It's just a process of learning because there's so much to it. So I put up two definitions that I found on the internet. One is application of statistics to a wide range of topics in biology, life science, community health sciences, and epidemiology, and many other related fields. A more general is a branch of statistics that deals with data relating to living organisms. So again, anything where you use some analyses, statistical analyses to analyze data that you collect and do research on. And the steps that biostatistics takes is, it's part of data collection pre and post. Again, you want to collect your data that it's statistically strong. So you want to guide the experimental design. You don't want to just blindly collect something and then at the end you're like, oh, I guess I didn't think this through. So it's kind of important to think about biostats before you start.
So you can't just like collect your data and then make your hypothesis?
No, I don't think that's a good idea. I mean, some people do.
It seems like it's fairly common.
Yeah, and don't get me wrong, like some people certainly do. Or in biostats, for example, is they'll look at data as like, oh, what have been collected? What could I ask? Like in health sciences, they collect your weight, your age, your height. What else do they take? They take your blood pressure every single time, right? So that is data that is regularly collected. So people go and I was like, oh, I'm going to take this data. Let's see what I could do with this. So that is one experimental setup in biostatistics. It's the cheapest one because it's already there and you do something, but it's also the weakest one because you didn't go in with a specific intent to do that. Then analyzing and interpreting the results. One question you have to ask is, are the mathematical and statistical calculations applicable to the real world issue? Sometimes the math is really clear, but does it actually relate to what I want to solve? And the math doesn't always model really well of what's going on in the real world. So it has to, you have to see if like, am I using the right math? Am I using the right statistics? And can relate that. It's not that easy. And then also proper statistical procedures. So one statistical procedure that's very popular to use is Pearson correlation coefficient. It has been used for years to do some correlation between two groups, for example. But it should not be applied to every data set because Pearson, in its development of the equation, assumes that your data is normally distributed. For a lot of times, your data is not normally distributed. If you can hypothesize that your sample data was pulled from a normal distribution, you can still kind of go with Pearson, but your data at hand is not really normal. If you have, so for Pearson, a normal correlation is like the bell-shaped curve, and if you have your bell and then it's kind of skewed a little bit, again, you can assume it's normal. It's not perfectly normal, but you can assume it and you can still go with Pearson. But if you clearly have two bumps you probably shouldn't be using Pearson correlation and should be using Spearman correlation, for example, to make up for the non-normality in it. So it's also important to check the other way around. Is your data allowed to use certain statistical procedures? And then at the end, what I mentioned before is the data representation. You can do it numerically, you can put some graphs, you can write an equation. So that again is sending stuff back to the original, because most of this is not all done by one person, because it takes different expertise to do everything of it. So that's kind of the gist of biostatistics. So and then there are different types of research areas too. There are different setups. For example, a cohort study is the most expensive one. It's the longest one. It takes a lot of dedication. You have to wait sometimes. I mean, they range from 3 years, which is a very short cohort, to 15 to 20 to 50 years. So sometimes you start a project and actually never get to see or work with the end project because someone else will pick it up. In the cohort you pick a group and then you follow them over the time. You normally pick, for example, smokers, and non-smokers is one of the standard examples, and then you follow them. And then you see, do they develop something else over time? Are they getting lung cancer more likely than the ones that are not smoking? Do they have premature birth? Kind of. So you can follow them and collect the data, but again, at the beginning, you need to be clear what data to collect. Sometimes throughout the time, depending on how they're developed over time, you're like, oh, I should have asked that question too, but it's too late at that point. So they're very long, they're very expensive, and they take a lot of time. Case control studies are often used for rare diseases. You pick a group that has a disease and you pick a group that does not have a disease, and then you figure out where the exposure came from. It's normally a small scale and has still some statistical, from the statistical viewpoint, it still has some power associated to it, although the case control are normally pretty small sample sizes. And the cross-sectional study, that's the one I mentioned before, is where you have the data and you try to figure something out from the data. So at that point, you don't know what got first. For example, if you checking diabetes and then you pull these records of heart rate, weight, height, you get it all at once. So you don't know at one point they're developed necessarily diabetes. You know they have diabetes, but you don't know at what point of exposure it happened. So they're very weak statistical indication studies, but you have normally a huge sample size to do a lot of statistics on. So that's just kind of the different areas, and there's certainly more. There are clinical trials. There are more types of research I think done, and then you can kind of check in which direction, what you have, how's it set up. So it's important to know what kind of research you do to do the right appropriate statistical method behind it. Another thing you really have to know first is what kind of data are you actually analyzing? Because for every type of data, you have different statistical methods. So the first thing is my data discrete or continuous? So discrete is like when you have a certain number of indices. So you have, let's say, number of children I would put into discrete because, I mean, at one point it stops, right? You have children. So on average, let's say between 0 and 5. I know that is not true, but that's kind of throwing in the average. So I probably would put that into discrete and not continuous. You also don't have a 1.5 child, right? So it's not kind of, you don't have a curve. You have kind of more of a scatter plot of how many children there are. Whereas continuous are measures that are taken over a specific range. So sometimes age. You can put age into continuous and discrete. Normally, the methods for continuous are much easier than the ones for discrete. So I would throw age into continuous just to make my life easier, because you are 24 and a half, right? Like, that is actually a continuous variable at least. Or your weight can be seen continuously. And then for these, we use quantitative numerical methods. And they're much easier normally to deal with, and they're more powerful than discrete. But if it's not valid, you shouldn't be using a quantitative methods if you actually have discrete variables. And then with discrete, you then need to differentiate, do I have a quantitative or a qualitative one? So quantitative, if you don't use age as a continuous variable, you can put age groups. I'm putting all the people between 10 and 15 in one group. Then I'm putting all the 15 to 22 in one group. So I'm building age groups, and then I'm using, let's say, the 6 age groups as my discrete quantitative variable. Number of children, again, what I mentioned before. So qualitative is categorical. So you don't really associate numbers to the data you collected. So a categorical could be binary. So binary is 2 outcomes. It's 0 and 1. You pass a test, but you fail a test if you're not talking about letter grades. Or if you marital status, yes, I'm married, no, I'm not married. So just kind of yes and no questions. These need to be analyzed differently from the other qualitative data, because you have different, you get something different back, so you cannot use the same statistical approaches. So you can also have ordinal qualitative data, and that is that you have a natural order to the data that you have collected. For example, letter grades. An A is better than an F. There is a natural order behind it. Or economic status, if lower income versus higher income, there is an order to it, right? There is an increasing income when you have a higher. Or what's often done too is that these ordinal qualitative data is transformed into quantitative numerical data. And you can do that because of this natural order that's in there. And that is often done just because these methods are a little nicer and they have more statistical power. So you can rank these. So if you use ranks, a letter grade A would maybe correspond to a 1, and a letter grade B would correspond to a 2. So then you can have some numerical values associated, and your analysis is a little easier. Nominal data, there is no natural color because I saw a color. There is no natural order. You have blue eyes, you have green eyes, you have brown eyes. You're still collecting the data, but you cannot pull an average from that, right? There is no order associated to it. So these are probably one of the hardest data to analyze or integrate into your data set. Birthplace is another one that is considered nominal, because there really shouldn't be an order if you were born in Reno or Sparks. It's just a given. You're born here or there, but someone born in Reno is not better than someone born in Sparks or vice versa. There's no order. Sex is sometimes treated wrong, I think, especially in our changing environment, because it is not just black and white anymore as much. Sex used to be often in a binary, and I don't know, especially if you do human studies, it's not necessarily true. It might be true for a specific study you're doing, but if you're looking at some gender transfer and how they're doing in life, and they do a cohort over multiple years, then that is probably not a binary variable, and it would more be a nominal variable. So these are the different data types. Based on what data type you have to use different statistical analyses. The easiest one I mentioned is quantitative numeric. The next one would be quantitative discrete. Easiest is continuous. The next one will be discrete, and then these will be getting a little harder and different methods need to be used. And then the data representation. So here I just have a small table of different people and the number of apples they're picking per minute. So you maybe want to analyze who should be fired, or let's investigate why is someone really good and why is someone not so great. So you can represent this data in different ways. A bar plot gives a measure for each individual measurement you're taking. So there's no order associated here. It just tells you Paul is picking 15, Scott is picking 19, and you just see the individual measurements that you took. A histogram gives you the number of people that fall within each block. So the first histogram has a length of 1. So how many people picked 9 apples? One person picked 9 apples. How many people picked 15 apples? Just one person. So it indicates how many people picked a certain amount of apples. So here, every bin is the length of 1 apple. And picking the bin depends on how you look at the data too. If, for example, I treat this, although I cannot pick 1.5 apples, if we treat it as continuous, the 1 maybe makes sense because I want to see at each instance what's happening. If I want to bin them together and look at them as a discrete one, where I smush the people together that pick between 5 and 10 apples, and then I smush the people that pick 10 and 15 apples. So we see that our histogram, I just noticed that it doesn't show everything. We smush them together, so our histogram will start looking a little different. So when you look at your data, think about how big you make these bins too, because you can change the look of your distribution quite a bit, depending on how you pick your bin. Then you can also use a density plot, which is really nice because it kind of does, give you an entire, like a smooth function. But again, density plots should be used only for continuous data and not discrete, because you don't have the spaces really in between. And it does portray, if you look at this right here, it doesn't really reflect the distribution of how people pick apples, right? And this is a really small data set. So density plots are great for continuous data, and if your discrete data are very close together, I think it's still okay. But when you start having spaces in between, it might not be the best way to go. And then we can look at the frequency. So if I look at Paul and I want to see how many apples did he pick from the total, he picked 11% of the total amount of apples that have been picked. So then we can also look at the relative frequency percentage on who is the best. So besides looking at the numbers, we see that Jake only picks 7% of the apples, so he might be someone on the list that gets a warning because he's not picking enough. Or maybe we figure out he was sick and he should have taken a sick day. You know, just kind of investigating your data. Another way to look at it is a box plot. And here you're doing a little bit more of descriptive statistics. So something we're all familiar with is the average and the mean. We're just calculating the total, and then we're dividing by the number of measurements we took. So here we have about an average of 17. The mode is the number that shows up the most, the measurements that showed up the most. And then again, if you have, for example, continuous data, the mode should be made into bins again, because if you have expression data, for example, you have 14.75, 13.73, 14.5, you will not necessarily have one data point that is exactly the same, right? So you maybe want to say, I want to put all the ones together that are between 13 and 14. So you would get more an actual information on what the mode would be. The standard deviation is calculated, I'm sure you're all familiar with it too, is how far are we on average away from the mean. And then we can talk about quartiles. The percentage of the number, how many are in the first quantile, 25% are in the first, and the next quantile is how many, another 25%, another 25% of the data, and then the last 25% of the data. So that's another way, and that's what the box plot does. It tells you the median rather than the average. So the median is the number in the middle of your data set. And if you order your data set. So here, I ordered the measurements in increasing order. So we're looking at the middle of the data set. We're moving in from each side until we hit the middle. And that would be our median. Our max and min is another information we can get from a data set. And then the quantiles, I said, it looks at the first 25% of your data, and if it's not even, it cuts in the middle and takes the average of the two values. And this is how the box plot is plotted. So we kind of get an idea of the distribution in a little bit different way compared to a histogram or density plot. And then we can also, a really standard approach, what I mentioned before, is correlating two measures. You want to analyze if there's any similarities between the measurements taken. So besides measuring how fast they're picking apples, we also measured how fast they're picking the strawberries, and we want to see is there correlation, are there as good of apple pickers as there are strawberry pickers in a way. I mean, it's a different movement. You're going overhead or you like on your knees underground. So then we plot, so each dot represents one person, how many apples they pick versus how many strawberries they pick. And then we want to see, do they correlate? We can eyeball it, but we cannot publish eyeballing, right? So we have to do some method to analyze, like we have to give it some meaning statistically. Instead of just saying, yeah, this graph looks good, we say there's a correlation, they relate to each other. So here I use both Pearson correlation and Spearman correlation, and we see that there are somewhat in the same room, and that is pretty common. And if you look at the two, the Spearman correlation coefficient is on a regular base more stringent than the Pearson, so your values will most of the time be a little lower. The reason why I also use Spearman is because this data is not really normal, and I cannot really assume that it is normal because people will be picking at different speed, but I'm not sure really how that will really, if it really will follow a bell curve or not. So if I would publish this, I would certainly be reporting the Pearson correlation more likely than the Pearson. And then so we see that the correlation is kind of weak, right? It's not really there. So if we would consider analyzing how they would be picking pears, if I would add one more column and would analyze how they relate between picking apples and pears, do you think the correlation would be bigger, greater or lower? I would assume greater too, just because of it's the same movement. You're doing overhead versus knees, right? So I would assume that the correlation between apples and pears would be greater as well. But because I just made these numbers up and it's not a real study, we cannot prove that. So another way to look at it, besides looking at correlation, is at a linear regression. So there are methods where you can just use a function, throw the two vectors on, and see is there a linear regression between the two. And the blue line shows that that is the best fit for this data set we have right here. But we see that none of these data points we have are actually close to that fit, right? So the linear regression we get an R-square value of 0.56, which is pretty poor. And what this is telling us is that a linear regression is not a good fit for this data. So we cannot really get a linear line that will explain this behavior between those two variables. So what this is saying in words, other than just having this R-square value, is that about 50 to 60% of the fruit pickers are closest to the best fit. So your linear line. So it doesn't talk about correlation. It talks about how close these data points are to the line. And when we look at this, we have 1, 2, 3, 4 within these green lines that are closest, and those would be our 50 to 60% of data points that are closest to this line. So the two methods answer kind of two different things. They're used sometimes together, but they really tell you different things. The correlation coefficients range between -1 and 1. So you can also, the correlation can tell you if they're anti-correlated. If one goes up, the other goes down in a perfect dependent way, then you would have a correlation of -1 with perfect anti-correlation. Or if they behave exactly the same, then you would have a perfect correlation of 1. And that's called correlation analysis. It measures positive and negative linear correlation between two variables. So how two variables correlate to each other, how are they related? Whereas the R-square value, the coefficient of determination, only ranges between 0 and 1. It's kind of a percentage of how close the data is to that regression line that you calculated on the data. So it's a little bit different. So they don't necessarily answer the same question, so they should be used in different situations. So that's kind of an introduction to biostats, kind of giving you an idea of what bioinformaticians do, how I got there, and what our center can offer. Any questions?
Thank you for talking.
Thank you for having me.
WNC. Thank you for talking about the difference between R and R squared. Because earlier this semester, as a group, we read a paper, and as we were going through it, we couldn't remember which was which. So the fact that you clarified that is very timely for us.
Thanks. I'm glad Laura gave me the hint.
Thanks, Julie. It brought a lot of memories from my PhD work. All the biostatistics. Yeah, I think biostatistics makes a lot of sense. When you teach it as a subject, it doesn't make any sense. But when you're doing your research and you have data and you don't know what to make out of data, that's when you learn to biostatistics, and that's the time when you start loving biostatistics.
Yes, I totally agree. I was sitting in class.
Unless you hate it.
Sitting in class and getting just what I showed right here with picking apples. It's fake data. You like kind of get the idea, but it's not fun. Unless when you get your project and you're stuck and you don't get to answer what you want to answer, and then you get to start playing, and then you play wrong for a while, but then you play right, and then you're like, yes, I made it. So it is a really rewarding and frustrating process, but it's fun. If you like the playing part, it is very much fun. And for one of my class projects, I worked on a melanoma project, and that was data I just pulled for my project, and I think that was one of the things I got the most out of that health community class, because I have a cousin who has melanoma, so I was more interested in it, on how it is, the death they do, and so having a related subject and then you get way more out of the class than just doing your homework every week when you are passionate about it. We're getting back to that, so that really helps. Yeah. Thank you for sharing.
I had a question.
Yes.
So, in your opinion, what was...
Do I look like a Smurf?
You look like a Smurf. Do you want us to say yes?
I don't know. I don't have a white hat.
No, you don't really have, but you are blue.
Yeah.
So, kind of look like it. But the orange letters, that really makes it look cool.
Yeah,
right?
there you go.
Anyways, so like for our level of research, and you know, we're doing research projects, we're collecting data. What would you recommend as a good statistical package? I just recently got a book on R for biology, or using R in the biological sciences. But I just wondered, like I used GraphPad, you know, before, and
I don't know anything about GraphPad, so I can answer to that.
Yeah.
But I've used SPSS and SAS in my classes before,
SAS is terrible.
and I think SAS is terrible. They have integrated function,
Oh my God.
but then you somewhat.
That was just, oh my god.
I used SAS for my PhD and you're saying it's terrible.
No, I
think it's very hard
terrible
to use.
Not that it's terrible.
I agree.
It does terrible
I
work.
agree with that portion.
Oh, it's a nightmare.
And
It's about
I think it is extremely hard to use. Where R is SAS free.
I don't think SAS is free. It wasn't whenever I used it. Rita would.
No, SAS is free. When I was at UNCC, it was free for the students.
Oh,
The
because
school
they had a
license.
license. But R is free.
R is free. You don't have to have affiliation to a university. You can use it time. And if you want to move outside of the university academic setting, R is also used in industry. So that's a kind of nice thing. SAS is used a little bit in industry, but not as much. I would recommend R, not just because I like using it, but it has a bunch of packages, and you can also make graphics right within your R environment. There are high-resolution graphics, so you can use them for publications right away too. Getting familiar with R will maybe take a little longer than SAS because it is command line. But to help out there's R Studio. That is a very nice interface, and they have a lot of tutorials on where to start and how to get. And then R just has tons of packages. So if you want to do a Kaplan-Meier curve to figure out the death rate, for example, they have functions for that. They have functions for linear regression. They have functions for multiple linear regression. They have, I mean, a dozen, and like people work on this continuously, and it's upgraded. And so I think R is
Worth learning.
Yeah, it's worth learning. Yeah.
Can I answer the question for GraphPad?
Yes.
Yes, I did use GraphPad, and I liked doing the diagram, like the graphs in GraphPad. It's very easy compared to SAS.
Yeah, I agree with that too. Yeah. Anybody else have any questions? No? Well, thank you.
Thank you.